Hello, my name is Amanpreet Kohli, and I am a PhD student at the Virginia Institute of Marine Science, where my research focuses on fish diseases. My lesson plan teaches high school biology students how to determine antimicrobial sensitivity. Finding out the sensitivity of microbes towards antimicrobial compounds, such as antibiotics, is essential to establish an effective cure for a disease. One standard method to determine the antimicrobial sensitivity is the Kirby-Bauer method, where bacteria are grown on an agar plate in the presence of antibiotics, and the results look like those shown in this image. The four circles in the center of the plate are antibiotic disc, and the clear zone around an antibiotic disc is called the zone of inhibition, which is formed due to inhibition of bacterial growth because of presence of antibiotics. The opaqueness on the rest of the plate is from bacterial growth. When there is no zone of inhibition around an antibiotic, the bacteria is rated as resistant to that antibiotic. And when a zone of inhibition is present, the bacteria is either completely susceptible or intermediately susceptible to that antibiotic. This decision is based on measuring the diameter of zone of inhibition and comparing it to standard values given in this table. As an example, for the gentamicin antibiotic here, the zone of inhibition measures 36 millimeters in diameter, which when compared to values here, show us that this bacteria is susceptible to gentamicin. In my lesson plan, I have set up an activity where students are helping me compare effectiveness of 4 different antibiotics for pathogens which are associated with lesions in fish. The setup for the activity: students will be divided into groups, and each group will receive 4 images which look like this. Each image corresponds to results from Kirby-Bauer experiment for a particular pathogen to test its sensitivity towards 4 different antibiotics. The group will record the zone diameter as well as the antibiotic sensitivity rating for the 4 pathogens and the 4 antibiotics tested. They will then compare the effectiveness of antibiotics within their group for the 4 different pathogens. All the groups will then combine their data. Each group essentially represents a replication of the Kirby-Bauer experiment, since all the groups are working with the same pathogens and the same antibiotics. Lastly, overall as a class, students will determine the effectiveness of each antibiotic towards each pathogen by calculating the percentage of replicates of each pathogen that were susceptible to each of the antibiotics. They will also represent their results in the form of a graph. I have provided questions for each part of the lesson plan to help encourage discussion. Additional questions are also provided at the end of the lesson plan, so students can learn more about pathogens and antibiotics. The students should learn about scientific experimentation from this exercise and the important aspects of scientific experiments, such as replication, collecting data, analyzing, and interpreting data. I hope you find this exercise engaging, and if you have any questions, please contact me. Thank you.